Hi everyone, welcome to another podcast of We Talk Science. Welcome. Today I have with me Dr. Britt Koskella. Hi Britt, how are you doing?
I'm doing well. How are you?
I'm really good. Thank you so much for joining us today. I was wondering if we'd take you through your journey of research into phage and plant interaction. Could you tell us, where you're from and what has your journey been till today?
Yeah, great. I'm excited to be here. So, my name is Britt Koskella. I'm currently an associate professor at the University of California, Berkeley in California. And I have been here running my lab for about 6 years. But before that, did my undergraduate, if we go all the way back then, at the University of Virginia. I was a biology major, but by no means a born scientist. I did not at that point or any time through my undergraduate career think that I would become a scientist. I then took a bit of time after my undergraduate degree as a research technician, a paid research technician. I was very lucky to find that job. And from there, I was hook, line, and sinker invested in evolutionary biology and ecology, which is my training. I decided to do my PhD in a lab at Indiana University working with Professor Kurt Lively. And Kurt had, among many fantastic scientific influences, one of his really impressive findings was that parasites maintain sexual recombination. So I'm going to quickly describe that because it's going to actually become formative for where I am at the moment. So Kurt had been studying this idea called the Red Queen hypothesis of coevolution. And this is the idea that by co-evolving with parasites, by running as fast as you can as a host population just to keep up with the evolution of your pathogens and your parasites, that you need to generate diversity. Okay? Because selection doesn't work if there isn't diversity upon which that selection can act. And so Kurt was using a model system of snails that lived in New Zealand. And in these lakes, there are snails that reproduce sexually and those that reproduce asexually, the same species, and they're both infected by this trematode parasite. And so he set out to ask a pretty simple question, which is, can sexual individuals, those that recombine their genomes and produce offspring that are not like them, are they going to be more resistant to parasites? Are they going to be able to evolve more quickly to escape parasitism? And anyhow, the answer after 20 or so years of research is yes, that is exactly what happens. You find, somewhat at first counterintuitively, you find more sexual recombination where there are parasites, because the parasites themselves are selecting for that sexual recombination. And it's only in environments that don't have many parasites that asexuals can proliferate. So I was working with Kurt, and for my PhD was doing work on this snail system, but wanted to do something that I had luckily had a chance to try as a research technician, which was experimental evolution. And this is the idea that you can create an environment in the lab or in a semi-natural environment where you control how natural selection acts. So in this case, we call it artificial selection because we are controlling what the pressures are. But you get to control what selection pressure is or is not there, and then watch in real time as these populations respond or don't through evolutionary change. And so I did that to test this idea I already introduced of the Red Queen theory of coevolution. And over the course of my 5-year dissertation, we had some tanks where we added parasites and some tanks where we did not add parasites. And we allowed the snails to evolve. The snail was initially a very diverse population of sexuals and asexuals, and we allowed them to respond to this parasite. And what we found is that exactly as you would predict under the Red Queen model of coevolution, the parasites adapted to infect and kill these common asexual lineages. So that what we call the common clone. It was essentially a sitting duck waiting for a pathogen that could infect it. And once that happened, for an asexual lineage, there's essentially no escape, apart from via mutational change, which takes a long time, especially for a snail. Everything takes a long time for a snail. And so just as we predicted from the field patterns, we could see in real time over the course of 5 years, this pathogen, this parasite, adapt to infect these common snail types and kill them and give an evolutionary advantage to rare genotypes. And I loved everything about that project apart from how long it took.
Yeah.
Snails, they have a long generation time. But I absolutely loved the idea of being able to choose the selection pressures, manipulate those selection pressures, and see how evolution or co-evolution between species changed through time. And so for my postdoc, I switched entirely over to bacteria and phages, these little viruses that infect bacteria. And in doing so, started my research career on working above ground on plant phylospheres. So leaves, bacteria and phages that live in and on leaves and flowers and stems, but mainly leaves. And I've been doing that ever since.
Oh, wonderful. So can I go back to the parasite question when we were talking about the snails and the parasites? So any kind of coevolution ideas that, that people have been looking at and talking about, there has to be some kind of a evolutionary advantage for the parasite to kill off the asexual snails and actually let the sexual snails basically reproduce and take over the population. What is that advantage for the parasite?
So remember that parasites are not forward-looking, nor is evolution. And so everything that happens can sometimes seem intuitively like the worst case scenario. So a pathogen doesn't know not to kill its host population, right? It doesn't know what would be in its best interest if we could, control things magically from above. And instead, what's happening is selection is favoring whichever pathogens reproduce more in the next generation.
Of course.
And so if there's a common genotype, that is any pathogen that can infect that common genotype of the snail is going to have an evolutionary advantage and be selected for, even though, as you said, in the long run, it's creating a dead end for itself because it's going to decrease the prevalence of its own, or the frequency of its own host type in the future. And actually, what you described just now is exactly how it works. And we call it negative frequency dependent selection. Mm-hmm. And it's the idea that parasites adapt to what's common. They drive those common things down. So they actually, in the end, drive themselves down.
Yeah.
And in doing so, they open up this space, this niche, these resources for other types of hosts and other pathogens as well, other pathogen types that can infect those other hosts.
That's really interesting because, I mean, when you talk about straight up evolution, when you see, for example, fungal infections, there are some fungal infections that essentially infect the host in such a way that it doesn't kill the host, but actually, allows it to proliferate even more. So we're talking about, zombie ants, for example, or these, I cannot remember the name of it, but there is a fungus that actually infects frogs that makes the males sing louder so that they, procreate the fungus, even more. So in that sense, are these fungi smarter in that sense in terms of coevolution, or they just figured out an advantage somehow?
So it's a great question. And we're talking about many different processes all acting at the same time. And so this idea of host manipulation, lots of parasites do this, host manipulation. And I'll come back to that in a moment. But in the absence of host manipulation, we do think a lot about how evolution should fine-tune pathogens. And it should not be that pathogens evolve to be as virulent and as harmful to their hosts as possible. That is not where evolution selects over time. It actually selects for what we call intermediate or optimal virulence.
Okay.
And so the reason is exactly that. Even though there would be a short-term benefit for a pathogen that really goes, kills everything and transmits itself a lot, as you said, the long-term evolutionary outcome would be selection against that type. And a more prudent parasite could have a longer success. And so there's a lot of theory to suggest that this sort of intermediate virulence is actually what's selected for. But as we're observing now with the novel coronavirus, that is over evolutionary time and after fine-tuning. Yeah. And of course, when you have these spillover events or you have these initial moves into new naive populations, it takes time for selection to act and get to that sort of final place, if such a thing exists. But going back to your point about, are they smarter? I mean, in some ways, not actually intellectually smarter, but of course, yes, intuitively, it is a better strategy if you can do anything to increase your own likelihood of getting to another host, of successfully reproducing, that absolutely will be selected for. But there are so many different ways to do that. One would be through host manipulation. Another one would be through, prudent killing, so not killing your hosts as quickly and shedding more pathogens into the environment. But yet another one, and going back to this idea of negative frequency dependent selection, would be if you're a pathogen or a parasite that can mutate rapidly so that you can keep up, then maybe it's not so big a deal that you're decreasing the frequency of one host, because some other host type will be around and you can just adapt and move over onto that host, which is exactly what happens.
Fair enough. It's quite complicated, because I guess, obviously, because the thing is that every species is trying to survive. This is the fight for survival, essentially. Yeah. But then I'm thinking that, and please correct me if I'm wrong, but microbes and pathogens are so numerous in number that they're willing to sacrifice part of the population in order to actually get forward, as a whole species. Whereas I guess, in mammals and, other bigger animals, that's not really the case, is it?
So really interesting question. And there's many layers to it as well. And I'm going to start with one of the more interesting layers and then go down into microbes, which is something that you said reminded me that, it's not just about sheer numbers. Their evolutionary theory includes something that's called inclusive fitness. Mm-hmm. And this is the idea that even if you yourself as an individual don't reproduce your genes, so you don't have offspring, if your being there increases the likelihood that your cousins or your nephews and your niece survive, they also have your genes. And so that idea that your fitness is not simply about you, it is about your entire lineage. Right. And of course, the place where that's been studied the most and where it's most obvious is in bee colonies, honeybee colonies, for example, where the vast majority of individuals never reproduce. They have a fitness of 0 if you just measure it as offspring. But it's not just offspring. The fact that they are there caring for the queen bee, who's just very related to them, right? And investing in the success of their siblings means that they have a high fitness, even though it's not the way we typically think about reproduction.
Interesting.
So going back to bacteria, though, they don't, of course, have caring or anything along those lines. But you're absolutely right that because they're genetically identical, you can have these huge populations of bacteria that the vast majority do not survive. Right. But nonetheless, if they're out competing another population of bacteria in a community, then that's an evolutionary advantage for that lineage.
Right. This reminds me of this wonderful experiment that was done by Roy Kashoni with the increasing amounts of antibiotic in the agar experiment, where you see this breakthrough of bacteria going into growing in higher and higher concentrations of antibiotic. Fantastic actually, experiment that was.
On that same note, because you mentioned population size, how many there are, the reason that you see that, if anyone hasn't watched the video, and I don't know if there's a way to share it on the webpage, but it's a fantastic video of a very large agar plate where the bacteria grow and then they stop. And then there's just this tiny little river, it looks like, of one mutation in a bacteria that then leads to this proliferation. Right. And of course, the probability that you have a mutant like that can break the resistance depends on population size. If there are more individuals, there's a higher probability of a beneficial mutation, because mutation, like evolution itself, is chance. Yeah. Mutations are almost always bad. They're always by chance, and occasionally, rarely, there's one that's beneficial. Mm-hmm. And the more individuals there are, when you have millions of bacteria, that's millions of chances to hit that right note of mutation. Yes. Yeah.
And that's why it happens so fast as well.
Exactly.
Yeah. Brilliant. Let's talk about phage and plants. So could you tell us a little bit more about what you're doing in terms of your experiments with the coevolution of plants and viruses?
Yeah. So I mentioned that I got to this point where I wanted to see coevolution in action, and I wanted it to happen faster than 5 years. Mm-hmm. So I started working initially on a plant pathogen, Pseudomonas syringae. It causes lots of disease in many agricultural crops, as well as horse chestnut trees in the UK, and now kiwi in Australia and in New Zealand and other places. And so it's a broad pathogen that people care a lot about. And it, like all bacteria, have these bacteriophages. The phages that I'm most interested in are the ones that we call lytic phages. So some phages live in the bacterial genome, and those are interesting for many other reasons. But what we work on are phages that don't live in the genome. They are what we call lytic, and they are obligate killers. So they bind to the outside of the bacteria, they inject their own DNA into the cell, they hijack the bacterial reproduction machinery, so there's no more bacterial reproduction. Instead, the bacterial cell itself becomes a sort of vessel for viral reproduction. And once the bacteria cell is full, it bursts out, and these phage particles, these viruses, are shed into the environment to go and infect new neighboring cells. And you might already be guessing this, but when you have something that obligately kills a pathogen, there's a lot of interest in it as what we would call biocontrol strain, right?
Sure.
So part of what my lab does is ask questions about if, when, and how we can use these bacteriophage viruses to stop bacterial pathogenesis, bacterial virulence, and disease, ultimately. And part of it is very straightforward. You can kill the bacteria. And that part we've known about since the phages were first discovered in 1920. That's why there was so much enthusiasm. That's why there are phage clinics still to this day in Tbilisi, Georgia, and across Russia, is we know that works. These are agents that kill bacterial pathogens. But we're actually more interested in the evolutionary implications of that. So there's also an alternative, and this should come as no surprise if you've been listening up until now, which is you can kill the bacterial pathogen, or you can select, you being the phage, sorry, I'm speaking in a funny way now, the bacteriophages can either kill the bacterial pathogen, or they can select for bacterial resistance.
Right.
Just like we see in that antibiotic plate. And what we're interested in is when that happens, which it will,
okay.
What does it mean? And can we use that selection pressure, phage-mediated selection, to our benefit? Once we've sort of accepted the fact that it's inevitable, that it's at least likely to happen, if not always going to happen, how can we leverage that to create better phage therapies and create better phage cocktails to control bacterial pathogens? And if you'd like, I can give you one quick snippet of how that works. Okay. So as an example, when our populations in a test tube evolve resistance to phage, they become less competitive. And this really makes sense because phages are typically attaching to receptors on the bacterial cell surface. That's how they recognize the bacteria. Mm-hmm. And one way to escape phage is to change that receptor a little bit so that you're no longer recognized. But that receptor is how it is for a reason. It's been fine-tuned by evolution to increase bacterial growth and fitness, right?
Right.
So when it changes, it's almost certainly coming at some sort of cost. We call it a cost of resistance. And it's what we see. Yeah. And so one cost of resistance is that our bacterial strains that have evolved resistance to phage, they are no longer as pathogenic or as virulent to the bacterial host. So they still grow on the leaves. They just don't cause disease. And this is incredibly cool, right? Because it basically means that you can use phage and have one of two outcomes, either of which is good. Either the phage kill the pathogen, great, or they don't because the bacteria has evolved resistance, but in doing so, it is now less of a nasty pathogen.
Okay, so essentially you're looking for both outcomes in this sense because, as you said, either it kills the phage, the pathogen, or it makes it less virulent.
Exactly.
Wonderful. So this is the particular pathogen that you were talking about. This is your model system, and has it been actually done in the field, or have you actually looked at it in a plant itself?
So we have not ourselves done it in the field. There are some groups who've started to use phage therapy in agricultural settings. And the short answer is it works sometimes.
Of course, because why should it be straightforward?
Exactly. But it can work. And I would say that Europe has sort of coined this term integrated pest management, this idea that we can't just we can't rely on single antibiotics or single chemicals anymore. Evolution is just too quick. And with the global spread of pathogens, diversity and mutations are just too broad. Resistance happens quickly now to almost any control measure. So what we need to start thinking about is a suite of control measures so that if one stops working, we have a plethora of other to fall back on. And I could imagine that's how I think about phage therapy as one part of a large arsenal to control pathogens. Okay, but to your question, we haven't done it in the field, but we've certainly done it in planta, in a plant. And the plants we use are tomato. And I'll tell you just two quick stories from the tomato phylosphere. And one of them is work done by Kathy Hernandez, who's a PhD student in my group, who's now just moving over to join Yale as a postdoctoral fellow to work with Paul Turner, keep working on phages. So what she did is what we thought was going to be a really simple experiment. You take a bacteria and a phage, this pathogen I've been talking about, and this lytic phage, that in a test tube, we can get the evolution of resistance in 8 hours in a test tube.
Whoa.
Okay? 8 hours, yeah. We put them in a leaf, and we wait for evolution to happen. We wait for resistance to evolve in a leaf. And she did this experiment, I don't know, easily 10, 15 different times in different ways with every parameter tweaked. And we never found bacteria to evolve resistance to the phage in the leaf.
Okay.
And this is kind of going to go against something I said a minute ago, which is that it's always going to happen. But of course, what I meant by that is it's always going to happen on a larger population level scale, right? Sure. When you're talking about hundreds and thousands of plants. But in a single plant, we never saw it happen. And through a lot of clever digging around and clever tests, she figured out why. And it goes back to all of the ideas we've just been talking about. It turns out that in a plant, two things are different than in a test tube. One is that there's spatial structure. So as a phage bursts out, it doesn't have this sort of mass, what we call mass action, where it has a chance to interact with every other bacteria in that liquid culture. Instead, it only has a chance to infect neighboring bacterial cells.
Okay.
And so a different way of saying that is that selection itself, selection pressure, is weaker in a plant. That's one side of it. But the other side is that cost of resistance I just told you about, where if you do evolve resistance, it's not selected for, it's actually selected against because you're outcompeted so rapidly by the sensitive ones that can grow better. And sure enough, those two things perfectly balance each other out so that even though mutations are certainly happening that lead to phage resistance, they are never being selected for. They're never increasing in frequency over time.
So what tips the balance then?
Well, that's a great question. I think two things. You could have stronger selection, so stronger phage-mediated selection, or larger bacterial population sizes, which also makes selection more effective. So again, if you scale up now to the size of a field, a whole agricultural field, selection is going to be far more effective at that scale.
Okay.
Or there could be a resistance that evolves that is less costly. And that is ultimately what we're all most worried about in a phage therapy setting, is not that resistance evolves, but that it evolves in a way that is less costly or cost-free, so that it spreads rapidly. And the same is true with antibiotic resistance as well.
That's really, really interesting. So, in this sense, you have it working in a test tube, you have it working on a leaf, I guess, in culture, but you need that sheer sort of volume of a field in order for it to actually work, in real life. Let's say, for example, as a pathogen. An antibiotic, sort of like killing a pathogen, essentially. Yeah, that's really interesting. So I guess you mentioned that these experiments have been done in the field, sort of thing.
Yeah, and again, it works sometimes with phage therapy in agriculture. One of the big hurdles, actually, most of the big hurdles, I would say, are not biological hurdles; they are technical hurdles. How phages tend to be quite UV sensitive. So just spraying them out onto a field, they don't last that long. They're pretty fragile organisms, all things considered. And other strange things, like if you put them into soil, they actually get bound up by the soil particles. And so they kind of get lost very quickly when you spray them on. And of course, the other thing is just to be able to grow them to that volume, to be able to apply at scale is a challenge.
Of course, of course.
Yeah.
Amazing. This is so interesting. I mean, I think I could talk to you about this forever. What a wonderful sort of way to think about real life problems. Using something which is so incredibly basic.
Exactly.
The phage has always blown my mind at how incredibly basic this organism is.
Yep.
Like literally on the fine line of living and non-living, and actually
Yep.
being able to use this for practical purposes is amazing. So let me just ask you one last question, which is a little bit different from what we've been talking about. What has your experience been, and how do you experience science as a woman? And if you had a undergrad, perhaps someone like yourself in the past who wasn't sure about joining research, what kind of advice would you give that person?
All right, great questions. So, in terms of gender and science, it's a tricky one. It's hard to generalize because, we only have our own one lived experience. And I was very lucky because I had a string of all male mentors who never once made me feel like a woman scientist. I was always, even from when I first got my first position just out of undergrad as a technician, it just never even occurred to me that would be something that would change how I engaged with science. But I was very lucky because it turns out that is not the experience of a vast majority, or at least a majority of women in science. And for me, when I did my postdoctoral work at the University of Oxford first, and then the University of Exeter, and at both universities, I became quite engaged in women in science issues and women in science initiatives. And that was the first time in my life that I really was faced with the hard data, where I was reading the papers, that you send the same CV to a panel of scientists and you ask them to tell you what you would pay this baseline technician, and you just change the name and the salary drops. Yep. You start reading more and more of these papers, and it was the first time just faced with the hard, cold facts. It was the first time that I realized that it probably had been happening to me and I just hadn't noticed. Ignorant bliss, you could call it. And that was actually a really difficult reckoning for me. Mm-hmm. Spending so much time thinking about it, I guess you could say I almost got jaded a bit. It became very challenging. And now I think I've come up the other side. But one of the harder things is, and this is true for me as a woman, but I know, for example, women of color, this is exacerbated tenfold at least. It's the problem that if you get an award, or you get a special speaking invitation, or you get a promotion, it's that question, not just the internal question, is this because I'm a woman? Is this because I'm a woman of color? For example, if you were, it's that second guessing your own qualifications, but then also knowing that other people are doing it. Did she get there because? Did she get that because?
Fair enough.
That's been the hardest thing for me lately. Yeah. And so I guess that goes to my advice, which is I absolutely love my job. I love being an academic research scientist. I love teaching. I love being able to mentor students. I think it's a fantastic job. And people are always going to doubt you and second guess you and undermine, your sense of self-worth, because unfortunately, even getting your papers rejected, getting your grants rejected, that is all kind of eroding self-worth if you let it.
Sure.
And so I suppose my advice is, maybe someone will think that you got that position just because X, or you got that grant just because Y. And, you could spend your time killing yourself thinking about that, or you could take it and do the best work that you can do with it and, count your blessings essentially and move forward and be genuine to yourself and genuine to the science you love. And I think that's the only way you can succeed, really, is just to put on your blinders and do your great science.
Yeah. That's great advice. Thank you very much, Britt. Thanks so much for your time. This has been such an incredible experience talking to you. Such an honor as well. And I really appreciate you explaining, the Red Queen hypothesis and the coevolution theories and things like that. So I'm hoping that I learned something. I'm hoping that our listeners did as well.
Wonderful.
So again, thank you very much for your time.
Thanks for wonderful questions.
No, thank you. All right. See you the next time.